I'm going to give a bit of a historical perspective, but in addition to that, I'm going to say a few words about the future as well as some of the opportunities that are currently available. How do we get to the first? I mean, how do we get to where we are, or how do we get to the mRNA vaccines? Phil Feldner and I just published a paper in Nature Reviews Drug Discovery, 60-year evolution of lipid nanoparticles for nucleic acid delivery. Needless to say, it's a pretty long story. So I'm going to cover this in a very a very brief manner. So in the 70s, this is when I was in Utrecht, as Ray was mentioning, I became obsessed by, this is the late 70s, by two basic properties of membrane lipids. One is that more than half of the lipids you find in a biological membrane, if you take them out and put them in water, they adopt these non-bilayer structures, hexagonal H2 phase. So pretty relevant question, why are they there and what roles do they have? The other one was lipid asymmetry, that the lipid composition on one side of a biological membrane is different than the other. And so again, what are the consequences of this in terms of the properties of membrane systems? And so these are pretty basic. This is really a plug for basic research because there was no intention here of developing vaccines or any other therapeutics. In '79, this was Mick Hope, we discovered that the presence of these non-bilayer lipids, such as phosphatidylethanolamin, if you put some of that, a few of those lipids into a bilayer forming membrane, then we found we could induce fusion. So the basic role of these lipids is to engender fusion, which is of course pretty vital to life in one form or another in biological membranes. So it also turned out, as I'll point out later, to be useful for the design of lipid nanoparticles that get out of endosomes. In the late 80s, we discovered that, and this is lipid asymmetry we're talking about now, that if we synthesized an ionizable cationic lipid, so a lipid that was, say, neutral at pH, a neutral pH, and was protonated, positively charged at, say, acidic pH of 11, we could induce lipid asymmetry in lipid vesicles if we had a pH gradient, so say pH 4 on the inside. The neutral form of these lipids can go across membranes very easily, go to the inside, get protonated, and then get trapped because charged compounds don't get across membranes very easily. And so we could drive these pH gradients, drive lipid asymmetry, and see various functional consequences. In the mid-90s, we discovered that PEG lipids, if we had them on the outside of a liposome, that they would inhibit fusion. On the other hand, if we had diffusible PEG lipids, then they could diffuse away, then fusion would then be engendered. And so, again, you can see how these tools, such as these diffusible PEG lipids, ended up being quite useful, a quite useful tool bag or toolkit when we were trying to put together the lipid nanoparticle systems. In 2001, we discovered that if we added negatively charged lipids to positively charged lipids, we could flip them from a bilayer to an hexagonal phase. And this really corresponds to the mechanism of action of cationic lipids. They're really able to disrupt, induce fusion with biological membranes. So again, fairly basic studies, but really turned out to be quite useful. But we got a bit distracted. So in the mid 80s, we found that we could load cancer drugs into these small vesicles. A lot of drugs are weak bases. A lot of cancer drugs, actually 50% of all drugs are weak bases, and they have the property that in their neutral form they can go across the membrane, get protonated, then they get trapped on the inside. And this is kind of an iconic figure just showing doxorubicin being accumulated to the point that it forms crystals on the inside of these liposomal systems. And so we founded a company in the early 90s to deliver cancer drugs more specifically to where they're needed in the body. But soon we started working on, this is me and 4 postdocs back in 1990. It's a pleasure to say I'm still working with these people so many years later. We have a very good team here in Vancouver. But we soon started working on gene therapies. We couldn't really raise enough money to put old cancer drugs into liposomes. Now, as you're all aware, in order to encapsulate nucleic acids in lipid-based systems, you really need positively charged lipids to get reasonable encapsulation efficiencies. But there are no positively charged lipids in nature. They're really quite toxic. And so we had to develop systems that did not contain positively charged lipids. What we found was we could use this DODAP, this ionizable cationic lipid that we'd made for the lipid asymmetry studies. We could load the oligos in this case is what we were working with, the nucleotides at low pH, and we found they were retained when we raised the pH to pH 7.4. And so we devised a process where we had the oligonucleotide in water, pH 4, a lipid mixture containing the ionizable lipids is indicated here, but also cholesterol and phosphatidylcholine and maybe a little bit of diffusible PEG. And then we could trap, the first thing to fall out of solution would be an inverted mice cell containing the oligonucleotide. To do this fast enough, you can trap them in what was termed a limit size vesicle or a limit size lipid nanoparticle. That's the smallest that's compatible with all the molecular components. And so this turned out this is a new lipid structure, kind of with a hydrophobic core. They can be long circulating, which is what we wanted to get to tumors, et cetera, relatively non-toxic, and really satisfying a large number of the properties that are needed for clinical application, including being scalable and reproducible. So this led to a collaboration with a company in Boston called Alnylam. In the 2000s, it was actually for 7 years we collaborated with Alnylam. And there the objective was to encapsulate small interfering RNA to deliver to silence genes in the liver. And so this was a remarkably productive collaboration. And we used a model, a model that turned out to be very useful. It's almost high throughput. We could do two experiments a week. We could check a lot of samples, essentially just injecting the nanoparticles containing sRNA to silence factor 7, and then assaying for factor 7 in the blood 24 hours later. Factor 7, of course, is made in hepatocytes and is a clotting protein. And so if we actually manage to silence factor 7, then of course we're going to see less factor 7 in the blood. And what we found was, and this was a really initiated by Marcus Cipollini here in Vancouver, who did a lot of the chemistry that's involved here. But what we found was that if we varied the pKa of the ionizable cationic lipid head group, that could very dramatically increase the potency of these systems. This is a log scale on the y-axis. And so you can see that there's a very sensitive, it's very sensitive to the pKa of the ionizable lipids. And we developed, over the course of the collaboration, we went from a ability to silence a gene in the liver, to silence factor 7, requiring actually more than 10 mg per kg. We were at 40 to start off with DODAP, and then and then ending up with something at 5 micrograms, so about 1,000-fold improvement. We have a therapeutic index of 1,000, so that we give a 1,000 times higher dose before we start to see toxic effects. So a very successful collaboration. Subsequently, Alnylam took this into the clinic to silence a gene in the liver called transthyretin to treat a disorder called transthyretin-induced amyloidosis. This is transthyretin is made in hepatocytes. It's secreted into the circulation. If there's mutations in the gene, then can form these amyloid fibrils that deposit particularly in nervous and heart tissue and cause very significant problems. There's no effective therapy. And so this is where you can see the rather nasty progression of this disease over the course of the 5 years. Very simple solution to this, what's obviously a devastating disorder, is that, well, if transthyretin is causing this disorder, let's just shut it down using a lipid nanoparticle sRNA formulation. This led to a phase 3 trial that was monumentally successful. The results were announced in 2017. Improvement in the neural impairment score was the primary endpoint. This is a p-value that's 1 over Avogadro's number. There's absolutely no doubt this drug works. And similarly for other secondary endpoints. So a curative therapy for what was a previously fatal disease, approved by the FDA in 2018. And so I thought this was about as good as it would ever get in my career in terms of being part of an effort that led to a result such as that. But we then went on, we being actually Acuitas in this case, to say, okay, well, if we can deliver siRNA, can we deliver messenger RNA? And so we do exactly the same packaging process to produce these systems. Worked very well. And what we found was we could make the liver make any protein we wanted. Of course, the liver produces 70% of the proteins in your body. It doesn't seem to mind making a few more. And this is an example for erythropoietin, where you can get supraphysiological levels of EPO following an IV injection and getting the liver, getting hepatocytes to make the hormone. So we were focused on this. There's a whole lot of obvious things to do if you can make proteins, make whatever protein you want in the liver. But we were approached in 2014 by Drew Weissman, who has been working with Katie Carrico, saying that we have a delivery problem. How can we get mRNA coding for viral proteins for a vaccine into muscle cells following an IM injection? And what we found was that this turned out to be a very good vaccine against Zika virus. And so this paper was published in 2017. Here we have messenger RNA coding for the Zika virus pre-membrane and envelope glycoprotein. So this worked extraordinarily well and led to a collaboration between Acuitas and BioNTech in Germany that was originally aimed at influenza, but turned into an effort to have to develop a COVID vaccine in 2020. And it ended up that the lipid nanoparticle that we supplied from Acuitas became incorporated into the Pfizer BioNTech COVID-19 vaccine called Comirnaty. All of you are familiar with the results that came from that, 95% effective for protection and with a reasonable safety profile. And then, as you're all aware, it had a great impact on ending or ameliorating the COVID-19 pandemic. Now, I'm just going to sketch through a couple of things here, and other therapeutics that really just the systems we have now are really enabling. And then I'll give a couple of slides on the future prospects and emphasizing some of the rational design possibilities. So I think the approach here, where we're able to do either replacement therapies, vaccines, or gene editing, really constitutes the next generation of pharmaceuticals. It makes a tremendous number of therapeutics possible. And these are now coming into the literature almost every day. It's remarkable to see. I mean, treatments for glycogen storage disease, basically mRNA coding for glucose 6-phosphatase, another rare disease, propionic acidemia, propionic-CoA decarboxylase. Have to go back to your basic biochemistry here. We can make all these proteins. Treatment for heart failure. This is involving an in vivo CAR T approach that Drew Weissman came up with, targeting T cells to fibroblasts that ameliorates a fibrosis. Cardiovascular disease, is a company called Verve in Boston, showing that if we gene edit PCSK9, we can knock down the levels of LDL in the circulation. This is kind of a one and done procedure, and it's essentially a vaccine against heart disease, which is of course a major killer. And these are amazing observations that we're making here. I believe that we're going to revolutionize cancer therapies with a combination of vaccines, in vivo CAR T therapies, personalized monoclonals, delivery of, say, tumor suppressor genes to tumors, et cetera, et cetera. All of these drugs, really personalized therapeutics can be produced in a matter of a couple of months. And that's, again, another as opposed to $1 billion in 15 years, these drugs can actually be made in real time and can be of direct use to a patient suffering from some incurable disease. So as of now, there's over 300, over 400 medicines in preclinical and clinical development, with more than 30 in phase 2 studies and beyond. Now, just a couple of words about the future. This is where in innovation we really do use a rational design approach as opposed to a high throughput approach to increase therapeutic index, access non-hepatic tissues, and although I won't talk about that too much, achieve tissue and cell specificity. One of the ways to improve therapeutic index is to make sure your mRNA is more stable, not as susceptible to degradation, either by interaction with the ionizable lipids or through other means. And what we've found here is if we segregate it by using, this is essentially inducing fusion at pH 4 during the formulation step to form these what we term bed structures, we can improve the stability of the mRNA with attendant improvements in the therapeutic or in the potency of these formulations. For long circulating systems, to really take lessons from nature here, erythrocytes circulate for 120 days. And our outer membrane consists of phosphatidylcholine, single myelin, and equimolar cholesterol. So let's make our our systems more like bilayer systems with that lipid composition. And what we found is that we can, if we have higher contents of these bilayer forming lipids, such as DSPC cholesterol, we can form these liposome-like structures with the mRNA and the ionizable lipids on the inside in the form of lipid droplets. And these systems have longer circulation lifetimes, and are, as you can see here, you can see the kind of morphology. And because of their longer circulation lifetimes, they can start to transfect tissues such as bone marrow much more effectively. And I think Dom will explain this further. So that's the end of my presentation. I just gave a bit of a historical presentation, just showing that really basic research, it was totally essential to this. We wouldn't have had the tools or the insight without that work. But really, the present day opportunities and the future opportunities here are really limited by our imagination. They really are, I think, transforming medicine. It's probably one of the most exciting times in medicine that's in history. So with that, I hope that I'll take any questions towards the end. And thank you very much.